So hello guys, welcome all of you. So today in this particular video, we are going to discuss about genomics data analysis for the beginners. So basically, as we all know that in this era where the genome data analysis is playing very significant role to understand the concept about the biological data sets, majorly if you are dealing with respect to the big data analysis, right? So here we are going to understand first what is meant by genome data analysis. So genome data analysis is nothing but where we are trying to work on the genome of the biological data, specifically the genes, right? And this genes has been encompasses with the data sets. Okay, it has been where the complex data has been stored in the one of the platform where we can examine as well as interpret those vast amount of data which has been stored in the form of the repositories. And why we need, guys, this kind of genome data to get analyzed is nothing but where we can all of you understand the basically the informations about the biological data. Agree? Okay. And those biological data, if you are able to understand, so there is a chances that we all can uncover the insights which is playing a very good role or the significant role in the biological data sets or the mechanisms we can understand. And that's why, guys, all of you can see that in this era where big data is being used for the biological data analysis, so ultimately the genome data is playing very significant role. Agree? Right? So in this particular context, we are all going to discuss in this particular video that how we can analyze the biological data specifically with the genome, and that data it's been used to understand the basically the type of mutations what we can detect, we can identify using the particular biological data sets of your interest, specifically with the genome analysis. Got it? So the genome is basically first we all have to understand, guys. So the genome has been comprised of the entity of the DNA, right? So basically it contains of the DNA sequence. So basically, and those sequences are in the form of coding as well as the non-coding region. Correct? And that is where we are trying to understand those particular DNA with coding and non-coding regions with the individual genetic blueprints are nothing but the makeup, right? So that's where you will understand the genetic makeup of an organism. And specifically, we can deal with the Homo sapiens as a particular species of your target, or else you can take help of other organisms to understand their genetic makeup or as the overall mechanisms with the target of your interest. Got it? So to unravel the hidden insights, as I mentioned earlier, so and to embed it in the genomic data. Embedded is nothing but which is stored in the form of the compact manner in the genome, right? So we are trying to uncover those hidden patterns, which is compact encompasses embedded in the genomics information. Got it? And that genomics information is specifically and sophisticatedly analyzed, stored in the form of bioinformatics platform. Got it, guys? So those bioinformatics tools are available where we can process the data using computational approach, and then we can extract the meaningful information from the compact data which has been stored, organized in the biological species, right? And that's why there is need, guys, to understand the genetic makeup of an organism. And the real example you all of you can take ahead is nothing but one of the pandemic COVID-19, right? So it's an outbreak. It is declared as an outbreak. So we need to understand the genetic makeup of that specific virus. So during that period, we used to understand the insights about the mechanism. So we have used the computational approaches. The researchers has used the computational approaches to uncover the insights because the data has been stored in the platform in the form of the repository. Got it, guys? And that's why we got a chance where we can uncover the hidden patterns with the complex data sets which have been stored in the embedded manner. Clear? Okay. So discussing ahead, what are the fundamental objectives of genome data analysis? What is the need, guys? Why we have to understand the genome data analysis? Okay. So it's really important. So why? Because it involves around diverse aspects of the genome. Okay, so there is a diverse information has been hidden. It's stored, basically hidden in the sense it's stored in the biological context, including the identification of the genes, regulatory elements, and also the functional components. Okay, so whenever if you're planning to identify the novel mutations, you want to identify the biomarkers throughout your research context. Okay, you want to identify the potential drug targets. You want to know the actual drug mechanism or is the pathways which is playing very significant role. Then one of the good approach what you can pick is the genome analysis, guys. Right, so there is a chances where you can identify the genes, the type of mutations, the variations, SNP mutations, and lots of things, right? And even link with respect to the functional interpretations and analysis. Do you think this is great, guys? Right? So yes, so that's why moreover to understand this comprehensive genetic variations as well as to explore the genetic relationship within the population or an individual. Okay, population in the sense where you can get to know the overall species information with the genome context, or else you can even concentrate a single individual genome, right? The two things we can do over here. So the significance of the genome data analysis has extended across a wide range of research discipline. Got it? And basically, it's encompassing the genomics, genetics, personalized medicine. That is exactly the future where we are looking ahead to design the drug targets and even to understand and study the evolutionary biology. Okay, and the very recent example which I have given is for COVID-19. Okay, so it is a one of the pandemic. So we are able to understand the evolutionary biology with exploring the genome. Agree, guys, right? And even the biotechnology. So why biotechnology? Because a vast amount of biological data has been experimentally validated. It's been studied using high throughput techniques and the tool by going with biotechnological approaches. So there is a high chances where the raw data has been processed. So using these concepts, we can explore the genome data. Okay, and then using the bioinformatics approach, how we can analyze the genome data, that is exactly we are going to discuss ahead. Clear? So here you can see that genome data analysis is rapidly evolving, as we all know that, right? For example, the NGS data, next generation sequencing. Okay, so it is playing a very significant or the vital role in understanding the genetic informations which is encoded with an organism of DNA. True, right? So genomics data involves the interpretations of vast amount of data. So whenever you can see, guys, that whoever the researchers are working in the field of genomics, they are basically using the NGS data. Okay, and if you discuss with any of the researchers, guys, it's very good manner, okay, where you can discuss and you will get to know the insights from the researchers. So definitely they will tell you that they are working with the big data, right? They are working with the complex data sets. So big data, complex data sets are nothing but the size of the data. Okay, the size of the data which is ranging from MB, 50 MB to GBs, like 1 GB, 2 GB, 3 GB, 30 GB of data. Okay, because ultimately we are handling the genome, right? So that's why the interpretation of the vast amount of data again linked with the genetic information is important. Okay, so this includes the identification of the genes, variations, and other functional elements which is related to understand the biological process, guys. Okay, and that goal we can reach. Okay, that is possible. Okay, and that's why there is a need to understand how this genomics data can be analyzed, right? So high throughput sequencing techniques are there, like NGS, next generation sequencing, which provides you the volume as well as the complexity of the genomics data in a very good manner to understand the overall pipeline, and then to necessarily to analyze that and using the specific methods and the tools again which is available in the bioinformatics platform. Got it? So then human genome is a big data. Okay, first you all have to know what exactly mean by human genome. Okay, so the human genome is composed of around 20,000 to 25,000 genes, which has been composed with the 3 million base pair. Okay, this is the important thing, guys. You all have to know this. If you're working with big data, if you're working with genomic context, right? So that is about 3 gigabytes of data, what you are handling. Now you understand, guys, what is meant by vast amount of data, right? So sequencing millions of human genome will add hundreds and of petabytes of data. Okay, that is a large size of the data, what I'm talking about. So petabytes of data, not I'm talking about MB, GB. Okay, it's around petabytes. It's reached till petabytes of data. Okay, and that data we can store, guys, in the bioinformatic platforms, right? Using cloud computing, using GPU servers, using the servers accordingly. Got it? So analysis of this gene interactions with multiple events we can study using again computational approach and analyzing the genomics data sets. So big data analytics becomes a critical in the genome because it is ability to store the data, transfer the data, analyze the data with a vast amount of genetic information. Okay, it's not like that we are just storing the data, but that data is relevant. It's contain a genome information about the specific species, organisms, or the target what we are interested. Okay, that is nothing but the disease of our target, right? So certain databases are there in bioinformatics platform where you can analyze the genomics data sets, such as GWAS, stands for Genome Wide Association Study, where it will give you a genome analysis and it will also analyze your data and exploring the connection between the genes and the target of your interest. Clear, guys, right? So moving ahead, key components of genomics data analysis. So the first is sequence alignment. So you have to know this because as you all know that the data is available in the form of sequence format, right? And for that particular context, we have certain sequence alignment tools are there which will help you to get to know the matching of DNA or RNA sequence with a reference of genome. Okay, maybe you are interested with the human genome or else any other species. So we have separate genome data sets available. Got it? Then the variant callings. Variants are nothing but the type of mutations, majorly the SNP kind of mutations, single nucleotide polymorphism, indel mutations, and so on. Got it? Then the functional annotations. You can identify the insights of functional relevances with the biological features. Okay, and then you can identify the pathways. You can identify the specific function about your target of your interest. Got it? Then the pathway analysis, that is what I have discussed right now. So you can identify the genes which is interacting and playing very significant role with the pathways. So that is really good, guys. Okay, in that context you will understand the actual molecular mechanism, biological process, and even the cellular process which is occurring in the specific process of the biological data, and again linked with the genome wide variations. Sounds good, right? So this is what you can do, guys. You can achieve the understanding with the genome analysis. Clear? So importance of genomics data sets. You all have to know this. Okay, if you understand what is the importance of this genomics data, then definitely it will give you a proper pipeline or the guide to plan your research with the computational approaches using genomics data sets as a data. Okay, so genomics data analysis have specific importance in the disease research. For example, you want to identify the genetic markers. There's nothing but the biomarkers, protein markers, drug markers, and so on. So you can identify based on the disease of your interest. And then those biomarkers you can classify like a genetic based on the diagnostic biomarker, prognostic biomarker, predictive biomarkers, therapeutic biomarkers, and so on, right? And then you can even look for the targeted therapy design. And remember, guys, this is the real research what it's needed. Okay, this is how you have to think about with the research point of view. Then evolutionary biology. You can study the evolutionary relationship between the biological targets of your interest. The next is pharmacogenomics. It's a connection, it's an integration about two different approaches. Pharmacogenomics. Pharma stands for the drug and genomics is specifically the genome data and analysis and interpretations. Okay, so using this clubbing or integrating these two approaches, you are able to identify the personalized medicine. Okay, you are able to understand the genetic makeup as well as the clinical phenotypic relationship between the drug targets, and that is really important, guys. Okay, the future is depend on certain kind of novel findings. So if you're able to understand how this particular steps is going on, so there is a high chances that you are able to look for the novelty throughout your research. Clear? Right? Then the agriculture genetics, again one of the good application of genomics data. Right? So environmental factors. Okay, you can study how you can improve the crop improvement, the disease resistance, and other agriculture applications, and so on. Right? And again, it has a very high demand, guys. Right? Then the forensic genetics, where you can analyze the criminal investigations, and then you will get to know the analysis of the DNA based on your known data sets which are available. Okay, that is really needed, guys. Okay. Then types of genomics data sets. So the genomics data sets are available in the form of the databases. Okay, so if you explore the databases of bioinformatics platform, guys, then you will get to know the types of genomics databases. Okay, like a DNA sequence where you will get to know the nucleotide information about the DNA strands. Then the RNA sequences are available where you are able to identify the RNAs, mRNAs, tRNAs, okay, ribosomal RNAs such as RNNAs. Okay, so this data you are able to fetch out with the genomic context, right? Gene expression data. Again, that is really a huge research is going on with the genomics data analysis using the gene expression data. So here there is a chances where you can identify the expression of gene patterns. Differential gene expression analysis. You will find a lot of papers which is get published on this specific topic. Okay, so gene expression analysis, and then you can even integrate with machine learning modelings also, guys. Remember, right? And then the epigenetic data analysis where you can analyze your data sets with the gene expression patterns, and then you will get to know the gene sequence with the mutations point of view. Then the next is the type of mutation is the structure variants, where you can study the structure variants informations with the sequence point of view. Basically here you are able to identify the large scale alterations as well as the link with the genome, like what are the insertion, deletion patterns, duplications, and the type of mutations which is occurring. Got it? So this is what exactly the type of genomics data are available. If you understand this thing, guys, then you will understand how you can reach the goals of the findings with the genomics data sets. Clear? Moving ahead, common genomics analysis tools are there. Okay, so why we need to understand which are the common tools are available or the resources are available to analyze the genomics data, because that will be ultimately a pipeline to analyze your big data sets, right? So genomics data analysis tools are used to extract the meaningful insights between your genomics data sets. The first is Galaxy. Okay, you can all of you use this Galaxy platform, guys. It is available where you can perform. It's an open source platform where it provides you a web based interface for data intensive biomedical research point of view, where it is integrated with a number of bioinformatics tools, and then you can process the NGS pipeline over here, okay, without need any extensive programming skills. Okay, so anybody who are working with NGS, definitely they have to switch or upgrade themselves with the command line coding. Okay, but if you want to make a trial how this NGS pipeline will work, next generation sequencing, then you can switch with Galaxy. Okay, the next is BLAST. I hope you all of you know what is BLAST database. So BLAST is a basic database available in bioinformatics platform, but again very important. Okay, it's where you can compare the sequence of DNA, RNA, and also protein to identify the similarity, and also what is the interfunctionality and as well as the evolutionary biology. Okay, how it is linked, guys, over here we have discussed about the evolutionary biology as one of the application. Agree? Okay, then we have IGV, that is integrated genomics viewer. So here you can view the genome based on the type of mutations, chromosome number, location, positions, and then an integrated viewer is there. Okay, you can identify the specific genes based on the genomics data analysis, and then you will download the formats to just represent the data with the genome viewer, and then ultimately it will help you to represent your research context. Clear? So these are very basic, guys. Still more databases and the resources are there, so you can explore with that context also. Got it? So the file formats, again important thing. Okay, there are different standard file formats are available to analyze your biological data. Okay, specifically with respect to the genomics data sets such as FASTA-Q, BAM, and VCF. Okay, so if you are working with omics data and specifically with the genomics data, so these are the standard file formats you all of you have to know. Okay, FASTA-Q stands for FASTA quality score. Okay, it will give you the score of your quality of your file, right? It's nothing but which contain the raw sequencing reads corresponding the quality scores about your data. Got it? The next is BAM, that stands for binary alignment map. It will give you and store the alignment of the sequence with the reads with a reference genome. So we have reference genome, guys, human reference genome 38, 37, okay, human genome reference 19 also available. So you can use that and then you can perform the analysis. Got it? So, and then it will store in the binary format where the algorithms can run of the NGS pipeline, and then you can work for the analysis point of view for the identification of variants ahead, right? Those variants are nothing but the VCF files. So VCF stands for the variant call format. So here you are able to identify the type of variations which is you able to identify based on the reference genome, and that is depend on the quality of the file what you are using to process your NGS data using genomics as an approach. Clear? Okay, so here you can identify the SNP mutations, that is a single nucleotide polymorphism, indel mutations, and there is again chances that guys you can identify the structural variations based on the functional interpretations. So ultimately you are able to study the overall process which is happening in the biological system, and that's why this genomics data or else the multiomics data is given very huge importance to identify the novel variations or to study the overall process about the mechanism. Okay, so understanding this format is critical, but data interpretation and selecting an appropriate tools of bioinformatics databases and analysis will help you to understand how the process will go ahead. Clear? So then next we are going to see some quick real examples of genomics data analysis so that you all of you will understand why there is need to explore the genomics data. Okay, what is the importance? The first is cancer genomics. So the objective, if you're working with cancer genomics, so you can identify the genetic or the genomic variations with the overall cancer development and the progressions point of view, right? And the methods what you can use guys with the bioinformatics approach is nothing but genome, whole genome sequencing, exome sequencing, variant calling, and even the pathway analysis. So if you use this kind of methods, so there is a chances that you can identify the biomarkers. You can look for the identification of personalized cancer treatments. Okay, so that is what you can do, guys. So remember, whatever the discussions we are doing right now, it is really important. Okay, then the infectious disease, you can identify the pathogens and the host responses based on the method of whole genome sequencing, transcriptomics analysis, and then the application where you can design the vaccine, study the epigenomic studies, and so on. Got it? Next is pharmacogenomic study. You can study the pharmacogenomics to identify the drug responses, and the methods you can use is expression profiling, whole genome sequencing, genotypic analysis, right? And then you can study the overall drug treatment with respect to the genetic profiling. Clear? The next is agriculture genomics, where you can study the how to improve the crop trial with a genetic analysis, and then you can identify the drug resistance mechanism and so on. Got it? Rare disease diagnosis, you can even work with the genomics point of view to identify the rare disease causing with the method of whole genome sequencing, exome sequencing, functional annotations, that is really important, and to treat the specific strategies for the application context. Clear? So that is all about guys with genome data analysis for the beginner context and majorly using the bioinformatics platform. So here are some recommended books guys for all of you to reference. So you can explore those and understand more about bioinformatics as well as the genome data analysis. So bioinformatics for beginners are there for gene and genome with molecular evolution. Very good book. The next is genomics for the data science, and bioinformatics sequence and data analysis is there. Got it? So if anybody wants to enroll for the to understand more about this kind of approaches, okay, in-depth analysis of the bioinformatics tools which are available, softwares, databases. So we do have certain internship is going on with Biotechnica. You all of you can explore the website, and then you can enroll for the coming internship program, that is bioinformatics summer internship program is there. Okay, and then if you're thinking then you do know about some of the omics data sets, and then you want to explore with some recent techniques such as AI, then also we have certain program is going on, internship program such as AI in biology and bioinformatics, where you all of you will get to know insights about how to build a model with the omics data sets, with the bioinformatics data sets, right? So please try to enroll for some internship program so that you will get in-depth hands-on session and experience to understand the concepts and implement accordingly with the research-based findings and also the applications and so on. So I hope that you all of you have enjoyed this session. So thank you all of you.